Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I'm Dr. Shahid Amin, and in this session we'll be discussing the second part of lecture 5 on biosynthesis of natural products, Chem 524. In this session we are going to discuss one of the major biological events occurring in living organisms, particularly in plants, and this is the event that is known as photosynthesis process. So let's see what are the topics that we have to discuss in this session. The topics include, first of all, an introduction to photosynthesis process, followed by the molecules that are responsible for this process that can absorb light energy, and then the phases of photosynthesis process like the light reaction and the dark reaction. First, we have an introduction to a photosynthesis process. Photosynthesis process is an integrated system actually in which light harvesting, photo-induced charge separation, and catalysis, these three processes combine to carry out two thermodynamically demanding processes. One of these processes is oxidation of water, and the other one is the reduction of carbon dioxide. So this whole process could be shown chemically as carbon dioxide and water molecules. They combine together in the presence of light absorbed by the light absorbing molecules. The major one is chlorophyll, and these carbon dioxide and water are converted to sugar molecule like in this glucose, along with the evolution of oxygen and small quantity of water. Now here we can see that this carbon dioxide, this carbon is completely in its oxidized form when it's converted to carbon dioxide. It will leave some of its oxygen and will absorb some hydrogen, so it will be a reduction of carbon. And here in this water molecule, this will lose hydrogen, so it will get oxidized. So overall the reaction is shown here. Now this photosynthesis process is a green plant process, and this is the means of converting light into chemical energy taking place within the organelles in the cells of the plants, and these organelles are named as chloroplasts. And these chloroplasts actually contain the photoactive substance that is responsible for this photosynthesis process, and this substance is known as chlorophyll. In this process, as we can see here, because oxygen is produced as a byproduct when this process is happening within the green plants, so this process sometimes is also called as oxygenic photosynthesis, that is the oxygen producing photosynthesis process, and this is the major source of oxygen in our atmosphere. In addition to green plants, some bacteria can also carry out the process of photosynthesis, and these bacteria contain a special type of chlorophyll that is known as bacterial chlorophyll. There are different types of bacterial chlorophyll, and the process of photosynthesis is carried out. But there is one difference, as in case of, for example, in case of green plants, we can see, as we discussed, oxygen is produced, but in case of bacterial photosynthesis, no oxygen is produced. The plant photosynthesis utilizes the solar light, and out of the solar energy, the visible light region, this is absorbed by chlorophyll so as to carry out this process. But in case of bacterial photosynthesis, they utilize only the infrared region of the electromagnetic spectrum of solar energy. The most important single metabolic innovation in the history of life on planet, this is photosynthesis process. And this is a process that is the most successful of all energy storage processes because it's happening for millions of years over the face of Earth in all the green plants. And this is also mother of all biological events of life, and it provides with all the free energy that is needed for the biochemical systems, and this energy actually originates from sun in the form of solar energy. And all form of life on our planet Earth, they have evolved to exploit oxygen for their well-being in the process of respiration. So they depend for their energy directly or indirectly on this phenomenon of photosynthesis. Okay, the light absorbing molecule, that is chlorophyll. Now we have some know-how about what type of molecule is chlorophyll. Chlorophyll absorbs light waves of all wavelengths except at the region 480 to 550 nanometer, and this region is known as the green window. And you can see here with this dark line that the spectrum for chlorophyll A, it's showing no absorption in this region, that is from 480 almost to 550, and this region corresponds to green light. This is the reason that the color of the leaves, it appears to be green, because this light is reflected back in our towards our eyes. So as a result, we see color green when we look at the green plants. Now, chlorophyll is of two major types, chlorophyll A and chlorophyll B. And these two types of chlorophyll, these are present in plants in a ratio of 3 to 1. If you look at the structure of chlorophyll, general structure of chlorophyll, for example, here we have a general porphyrin structure. Porphyrin is a pyrrole containing cyclic system like this one. Here are actually 4 basic rings: ring A, ring B, ring C, and ring D. Ring E is an extra ring here in this particular structure of chlorophyll. And in between this porphyrin, there is a magnesium 2 positive ion that is bonded with these nitrogen atoms. And so this special type of porphyrin, this is known as magnesium porphyrins. So chlorophylls are magnesium porphyrins. In its structure, there are some salient features like here are present these groups along the periphery of this system, and there is a group R that is in the form of ester, and actually this is derived from phytol, that is a long chain 20 carbon alcohol, and it gets esterified with the main body of chlorophyll molecule. And here at R1, it gives rise to different types of chlorophylls. For example, if R1 is methyl group, then this is chlorophyll A. And the chlorophyll A UV spectrum is given here. If this R1 is CHO, that is aldehyde group, then this is known as chlorophyll B. And because aldehyde group results into elongation of conjugation, so the absorption will be shifted towards red light, towards the higher wavelength. So its absorption is at lower energy or higher wavelength as compared to chlorophyll A. If you look at the major band that is towards the left side of this picture. Now, if you look at the bacteria that can carry out the process of photosynthesis, we see that in case of bacteria chlorophyll A, this group, that is CH double bond CH2, this is replaced by COCH3, that is an acetyl group. So in both chlorophyll A and chlorophyll B, in case of bacteria, this group will be replaced by an acetyl group. But when we look at ring B is further modified. Its modification is that in case of chlorophyll A, bacterial chlorophyll A, instead of this system will be like this. That is, this double bond, this will not be there, but these bonds, these will be as such. Instead of this double bond, this will be a saturated system now at this position, this between these two carbon atoms, and here we have this degree of saturation here. The two hydrogens are present here. In case of chlorophyll B, this is further modified, this ring B. Instead of having this CH2 and CH3, that is ethyl system, it has a unsaturated system. That is, the carbon of this pyrrole ring, this gets it has unsaturation along with the other carbon that is of this ethyl group. So this group is modified to this one. So these two are the structures for the bacterial chlorophyll. Now, if we look at the process of photosynthesis, that how does this process of photosynthesis overall it happens, then we have that we see that there are two major sets of reactions. One set of reaction is known as light reaction. This is named so because this depends upon light, and the other one is dark reaction or Calvin cycle. This set of reaction, it doesn't require any light, but this is dependent upon the light reaction. This is subsequent reaction, not a single reaction. Actually, these both of these are sets of reactions. So dark reaction, this is dependent upon light reaction. So when we look at where do these sets of reactions happen, as we discussed before, that process of photosynthesis happens within the green plants. Within the cells of the green plant, there are specific organelles that are known as plastids, and out of these plastids, the green colored plastids are known as chloroplasts. Inside chloroplasts, this whole process happens. So the machinery that is actually responsible for photosynthesis process, this is known as the grana or thylakoid. We will discuss this further. So this light reaction happens within these thylakoids, whereas the dark reaction, it happens within the stroma, that is the main body of these chloroplasts. First we look at the light reaction. The antenna proteins that are actually responsible for capturing sunlight, these contain many chromophores like chlorophyll, and some other chromophores also there. So out of these chromophores, chlorophyll A is the central pigment that is responsible for photosynthesis process in green plants. So this is the structure of the antenna. This is a manifestation of antenna proteins. Here you can see this blue color. This is antenna protein, and these green color and red color structures, these are actually the pigments that are responsible for capturing the light. This protein, this antenna protein, along with the dispersed pigments, this is present at specific structures. If you start from the structure of the cell of a plant, inside the cell there are plastids, green colored organelles. If you look at the details of the structure of plastid, it's a bimembrane system, and inside this body there are stacks of different discs. These discs are known as the thylakoids, and the stacks of these discs are known as granum. One is granum, and if we talk about many of these are grana. These grana are interconnected by these tubules that are actually the extensions of these membranes of these small disc-like structures. So disc-like structure is not a solid structure, it's hollow inside, so there is a lumen inside. The outer part is stroma, that is the main body of these plastids, whereas inside the thylakoid there is the inside body of thylakoid, or this is known as thylakoid space. So inside these membranes there are present the antenna proteins, as it's shown here. For example, this is one thylakoid, and if we look at this part of the thylakoid, so this is in detail part of detailed structure of thylakoid, and here we can see these are the proteins that actually have attached to them these chlorophyll or other chromophore molecules that are responsible for capturing the light. So the trapped electromagnetic radiation of the sun causes the flow of energy within the photochemical apparatus containing specialized reaction centers, allowing the trapped energy to participate in a series of biochemical reactions. And these biochemical reactions, actually, they are the part of this light reaction. So the story starts from inside these at the membrane of the thylakoids. For example, here this is a thylakoid, and it contains, there are two main systems that are responsible for capturing the light energy, and these are photosystem 1 and photosystem 2. So the biochemical reactions that happen here in this whole structure, these actually start from photosystem 2. When sunlight it falls upon this system captures this light, and water molecule gets split up into atomic oxygens that then combine to give molecular oxygen, along with protons and electrons are ejected, and these electrons are accepted by this photosystem 2. This photosystem 2 then transfers these electrons to plastoquinones, specific bodies that take up these electrons, and they take up protons from the outside of this thylakoid, that is from the side of stroma, and they take up these protons. These protons and electrons are coming from photosystem 2, and these electrons are further transferred to cytochrome, and from cytochrome they are transferred to plastocyanine, and then to photosystem 1. Photosystem 1 then absorbs energy, and these electrons are further transferred to some more systems. Ultimately, these are taken up by NADP that takes up protons and is reduced to NADPH. The protons that were released from water molecule, these are accepted by ATP synthase system, that is an enzyme system that will convert ADP to ATP. And these protons, these are channeled from inside of the thylakoid lumen to outside system, that is the outside part of these thylakoids, and that we call as stroma. So these protons are transferred outside of these bodies. So because of presence of a number of conjugated double bonds in the chlorophyll skeleton, much less amount of energy will be needed for carrying out these processes. When we talk about the first singlet state of the system, the second singlet state requires somewhat higher energy, that is energy 2 in the blue region of light. Now the excited chlorophyll as such directly cannot transfer the energy to a right location where the ultimate reaction will be happening. Rather, it initiates this electron transport chain that we have talked about now, and this whole system actually works in coordination, and as a result we have ultimately we get dissociation of water molecule into oxygen and protons, and formation of NADPH, as well as formation of energy-rich molecule ATP. Now we talk about the second phase of photosynthesis process, that is the dark reaction. This reaction was studied in detail by Melvin Kelvin in 1961, so it's named after Melvin Kelvin. So this process actually, this is the fixation of carbon dioxide. We know that carbon dioxide is the essential starting material for the process of photosynthesis. So this carbon dioxide is fixed by this cyclic process. Now, during this process, reduction occurs, and pentoses, that are 5-carbon sugars, are formed in a cyclic process. So the process is also known as reductive pentose phosphate pathway. Calvin cycle consists of three stages. These are fixation of carbon dioxide, reduction, and regeneration of the acceptor. Now these phases will be discussed a little bit in detail. Fixation of carbon dioxide occurs when a 5-carbon sugar that is known as in its phosphorylated form, D-ribulose 1,5-biphosphate takes up 3 carbon dioxide molecules in the presence of a specific enzyme named as Robisco, and this process will be a carboxylation process because as a result there is formation of a carboxylic acid molecule. So with the combination of 3 molecules of this compound with 3 molecules of carbon dioxide, we'll be having 6 molecules of D3-phosphoglycerate, that is a 3-carbon phosphorylated sugar. It will be converted to its biphosphorylated form by taking a phosphate group from ATP molecule. So for 6 molecules of this compound, 6 ATP molecules will be required. So this is up to now, this is the first step, that is the fixation. Afterwards, this fixation leads towards reduction, that is this glycerate, that is a ester of carboxylic acid, phosphate ester of carboxylic acid. It will be converted to an aldehyde. So from carboxylic acid towards aldehyde, this process is known as reduction. We know that if we go from alkane to alcohol, this will be oxidation. From alcohol to aldehyde will be oxidation. From aldehyde to carboxylic acid, it will be oxidation. But if we carry out the reverse process, that is from carboxylic acid to aldehyde, and from aldehyde to alcohol, and from alcohol towards alkanes, then this process is a reduction process. And the reducing agent that will take part here, it is NADPH. So both of these molecules, these were generated in the previous part of photosynthesis process, that was the light reaction. So these are utilized here. So reduction of this carboxylic acid will happen in order to produce this aldehyde. Afterwards, there is a series of reactions that will lead towards formation of, again, the acceptor species, acceptor of carbon dioxide, that is D-ribulose 1,5-biphosphate. So it's produced in this long series of reactions. During these reactions, a specific something special happens, that is the actual goal of photosynthesis process, that is to prepare food for the plants, and this food is in the form of glucose. This glucose is produced here as glucose 6-phosphate, and this molecule later on by dephosphorylation process will be converted to glucose, that is the primary food that is produced by this photosynthesis process. Now we talk about some important points about the dark reaction of photosynthesis. In this process, we have seen that there are several molecules that are generated, and they contain 3 carbons, they contain 5 carbons, they may contain 4 carbons, 7 carbons. So these molecules play an important role in the biosynthesis of natural products. Phosphoglyceraldehyde, that is produced as a result of reduction from its oxidized form. It serves as the mother molecule for the other sugars that are 4, 5, 6, and 7 carbon sugars, as we have seen in the previous slide, including carbon dioxide acceptor molecule, that is the molecule number 1, ribulose 1,5-biphosphate. Most of the D-glyceraldehyde 3-phosphate is recycled to D-ribulose 1,5-biphosphate. That is the main, that is the acceptor molecule, acceptor for carbon dioxide fixation. The remaining four, that is this molecule, will be used immediately as a source of energy, as we know that this molecular active ingredient or active intermediate in the process of glycolysis, and glycolysis process is uh, anaerobic oxidation of glucose. So it's an energy producing process, so it will produce energy in that process. The part of dihydroxyacetone phosphate which is not used in Calvin cycle leaves the chloroplast and can be degraded via glycolysis to provide energy. 3-phosphoglycerate that was converted to phosphoglyceraldehyde by reduction process. The second phase of the second reaction of the second phase of dark reaction. It gives rise to pyruvate that at the primary metabolic level, that is during the glycolysis process. 3-phosphoglyceric acid or glycerate may also be formed by breakdown of D-glucose via D-fructose 1,6-diphosphate, that is the general glycolysis process. Pyruvic acid itself is a very important key compound. It forms acetyl coenzyme A that enters into the formation of fatty acids, polyketides, and aromatic compounds, and also terpenoids and steroids via mevalonic acid pathway.